Sterile environment. It was all about washing your hands. I just hated my mom. You know, I didn't want to bring kids into my house because we couldn't be kids, you know, sort of thing. But then, you know, in industry, I'm amazingly impressed by bacteria, and as a biologist, I'm amazingly impressed by bacteria because, from my standpoint, they're way, way more evolutionarily advanced than us, because they can reproduce basically every 20 to 30 minutes. It takes us 20 years to have a mutational change. So, you know, like, who's more advanced here? And clearly, bacteria are withstanding frontal assaults from the drug industry, and yet able to develop resistance to these frontal, you know, if you will, nuclear bombs against them. So, but nonetheless, we still have this viewpoint, you know, that bacteria need to be eradicated. I can't really see from here what the heck I wrote on the slide, so it's kind of nice to see. And we need to protect not only ourselves, but the food supply, because you're hearing all this stuff, you know, chickens, beef, what have you, milk, you're going to learn more about, but, you know, how do we protect the food supply from bacteria? So at the end of the day, you know, we think we're going to save lives, improve lives, cure people by continuing to eradicate bacteria. But, you know, what I'm going to do is pass the presentation on to my colleagues here that have another sort of angle and a more holistic viewpoint about the microbiotic environment in which we share. So I'll introduce Colleen Cutcliffe, and you can introduce yourself briefly.
Hi, I'm Colleen Cutcliffe. I'm the CEO and co-founder of a company called Biome, and we're developing therapeutics that target the microbiome. So this is kind of a nice small group here. I'm just wondering if we can get a survey from the audience. Who here knows what the microbiome is? That's awesome.
Wow.
That's
wow.
awesome. Okay. A year ago.
Those are good questions.
Yeah. A year ago, there would be like 2 hands raised, so that's awesome. Well, the microbiome really refers to all of the bacteria and fungi and viruses that exist on the outside and the inside of you. And, you know, I think where we hopefully will be able to inform you guys and educate you on some new things today. This sounds like a pretty educated audience to begin with. But the first thing that we always talk about when we talk about the microbiome is kind of challenging this concept that, you know, we are not the center of the universe. And this is something that we are always coming back to as a race, because we are constantly thinking we're the center of the universe and being challenged. The Earth is not the center of the universe. We as humans are not the center of the universe. Silicon Valley is not the center of the universe. It's a hard pill to swallow, but it's true. And in fact, we're part of this really large tree of life, which is comprised of far more than just us. And so this is a little map. Mohan actually made this. I love the map. You are here. And I think one of the important things that we hope that people come to appreciate is that you're not sort of this entity that is separate from all the things around you, but you, your environment, your pets, your friends, your colleagues, you are in constant contact with the world around you, and you're constantly transferring your microbiome to other people, and other people are constantly transferring their microbiome to you. And so you really are one with the universe. And what we're starting to realize is that when we think about health and medicine, that we shouldn't think of people as a single entity or a gene as a single entity, but in fact, there's this very holistic picture about how we interface with our environment and how our environment interfaces with us. That's plays a very strong role in medicine. And the the concept of microbiome medicine or therapeutics or diagnostics that target the microbiome is really taking this concept and trying to figure out how we can intervene in ways that take the entire whole picture into consideration. And I'll just take a moment here because this is such an important concept in how we think about the microbiome to see whether my colleagues on the panel here have anything they want to add.
Well, I think there is this notion that's been emerging about the microbiome as sort of a forgotten organ. Like, yeah, it could be 2 or 3 pounds of mass in a human body, so it could be figured that way. But I think, as Colleen said, I think that perhaps a better way to think about it is it's sort of a connective tissue that not only is inside of us or around us, but it also connects everything that we come in touch with. Our pets, of course, but also the food we eat, the soil under the food we eat, the animals, both in the farm as well as in our homes. So it's sort of a connective tissue, thin tissue of bacteria that we live in. So we kind of grew up in it, and they were there before we were there. So we got to adjust and sort of figure out how humans can evolve underneath that umbrella.
Mm-hmm. Yeah, I feel like as humans we really like labels. We like to apply labels of good, we like to apply labels of bad. But the microbiome, it's not that simple. So next generation sequencing technologies can tell us what bacteria are present, but it's up to us to decide if those are beneficial or not. And it actually requires more than a species level identification to know if a particular bacteria is good or bad. So we have staph on our skin colonizing us all of the time. It's actually only very infrequently do you see an infection. So here we have an example of a bacteria that can act as a conditional pathogen. Under most circumstances, it might be completely innocuous. Only under a certain set of conditions, it can cause problems for us. So that's one of the things that as we think about the microbiome, we have to be very careful when we start deciding, what's good and what's bad and for who. Because the situation is even more dramatically different for patients who are immunocompromised, for example.
So, and I just wanted to add, so going back to, again, my academic days and training, it was all about rats and mice, right? That's what you earn your PhD in. And, what I eventually got was this book called The Vertebrate Story. And for me, I kind of realized that we're just this weird type of fish that happens to live on the land. But that there's really many, many, many more organisms out there than just the vertebrate, animals. And then I look at the bacteria, and it impresses me even further in terms of the diversity out there.
So I think when we think about the microbiome, one of the biggest hurdles in this field is thinking about all of the data that's out there. So just to give you a little bit of a sense of scale, we think about human genetics and what a huge problem we've been analyzing and housing and trying to make heads or tails of that data. We talk about the microbiome, now multiply that by 10, 100, 1,000, and then you're trying to combine that with human genomic data. And so this is at the very heart of a data problem, a data analysis and a data management issue. I did want to give a sense of where we are in this space in terms of knowledge, and one of the ways to do that is through publications. And so this is a figure that Bernard Ole published, in which he was looking at the number of publications and patents in the microbiome space. And on the x-axis is year, and the y-axis is number, and in blue are the number of publications every year, and you can see that they have been growing, and then I just sort of added in, this is the number of publications in 2014 that were made on the microbiome. Definitely a growing field. The amount of knowledge coming out of academia and now out of industry is continuing to grow at that rate. And then to give you a feel of some of the things that have been very instrumental in propelling the knowledge in the microbiome space include next-gen sequencing, which has allowed us to really profile microbiomes, as well as cloud computing, which allows you to have a place to put the data and then retrieve it. So this is a growing field, but I also want to give perspective on how much you really know about the microbiome. So in 2014, this is the number of publications that were made in the microbiome. This is the number of publications that were made in breast cancer. And this is the number of publications that are made in diabetes. So it's a growing field, but it's definitely a new field. So everything that you know and everything that you're learning, you're really at the cutting edge and will likely change as we get more knowledge. So we want to start with one of the first things that I believe probably everybody in this room has had experience with. You have all probably taken an antibiotic at some point in your life, and in doing so, you've completely transformed your gut microbiome. So you took an antibiotic because you had an infection of a specific bacteria you're trying to kill. What you did inadvertently after you took that antibiotic was to kill all of the microbes in your gut. And this is a diagram just sort of trying to show you what happens in the gut. You start out on the far left side with a nice ecosystem of a bunch of microbes that exist in your gut. They play a large role in things like metabolizing the food that you're eating, interfacing directly with your immune system. And when you take an antibiotic, you wipe out pretty much all of those microbes. And so a couple of things happen here. The first is that most of your microbes are eradicated, but the second is that some of them aren't. And one thing that can happen is that something that is actually usually at a very low level of existence in your microbiome, in your gut, can now all of a sudden propagate unchecked. It has no more competitors around it to keep it down. And so this is the nature, if anybody's heard of these C. difficile infections. This is what happens. You take an antibiotic, C. diff exists in everybody's gut at very low levels, and what happens is that that C. diff is allowed to propagate. And when it propagates unchecked, this causes a lot of adverse reactions. So getting back to Nick's point, in the context of the ecosystem of the gut microbiome, it's no big deal to have C. diff. In the context that there's nobody else around, you're now facing death. So the second thing that happens is that after a while, your gut microbiome will repopulate itself, and this is through environmental exposures, the food that you eat. And what people have noticed is that when your gut microbiome repopulates itself, it doesn't always look like it was before the antibiotic treatment. So why does this matter? Well, antibiotics have been used for a long time by farmers. Farmers have been giving antibiotics to their cattle for decades, not really understanding or thinking about the microbiome. Why have they been doing that? They've been doing that because they noticed that when they fed their cattle antibiotics, they gained weight. And so for anybody here, you can get more out of your cattle, per cattle, this is great for business. So they've been feeding antibiotics to their cattle, noticing they gained weight. And one of the things that we have found is that this doesn't just apply to cattle, it applies to humans. So Martin Blaser published a paper in which he looked at 16,000 children some of whom were on antibiotics systematically, and some who were the healthy controls. And what he found was that kids who are systematically on antibiotics are more obese than kids who aren't. And that perpetuates throughout the rest of their lives. And so when you take antibiotics, you're not just killing some microbes, you're not just changing your gut microbiome, you are really changing your metabolic state in ways that we are just now starting to understand. And so one might consider all of that to be correlative information. Okay, you take antibiotics, it changes your gut microbiome. You take antibiotics, you gain weight, but does the microbiome change really cause the weight gain? And that was answered actually in this landmark study, which has been repeated by multiple groups, in which they took this obese woman, they took her gut microbiome and they transplanted it into a thin mouse, and that thin mouse became obese. They then took this thin woman and they transplanted her microbiome into an obese mouse, and that obese mouse became thin. Then they took the microbiome from this thin mouse, who had gotten the microbiome from the thin woman, and they put it into the obese mouse that got the microbiome from the obese woman, and what do you think happened? That obese mouse now went back to being thin. And the really cool part about this study is that these two women here, this obese and thin woman, are genetically identical twins. And so what that means is that all of this weight gain or weight loss is completely being driven by the gut microbiome. And this is where there's the promise for the beginning of new therapeutics to that intervene and change the microbiome to be able to change metabolism. And this is just one example that I think everybody can identify with, is familiar with. There are a ton of other ones that continue to come out, and we'll hear about some more of them today. And this is talking about the gut microbiome, but the skin microbiome is another place in which you are constantly changing your interface with the environment and your immune response. So just to take a second to think about that, you have co-evolved so that on your skin there's a layer of protected microbes. And when I say protected, what that means is they provide a barrier between the allergens in the world around you and your host immune response. And what we are doing systematically, some of us multiple times a day, is stripping that microbiome, that protective barrier, so that now your immune system is having to deal with the barrage of every single insult and antigen that is out there and responding to it. And so when we wonder about why allergies and asthma and all of these immune responses are going unchecked and out of control, is because we've removed a barrier that's normally keeping allergens away from our immune system. And when we remove that barrier, we are causing our immune system to have to be in constant battle mode.
So to continue a little bit with that, I was thinking about the amazing changes or disruptions. I've been hearing a lot about disruptive technologies in this conference, but the amazing disruption that we've done to our microbiome in the last, say, 150 years. 150 years is a very short amount of time, evolutionarily speaking. But if you think about it, we've done at least 3 categories of actions. We've changed the birth process, and I think those of you who've been in this process, been in this literature, know that C-section babies and vaginal birth babies have a vastly different microbiome. C-section babies resemble the skin of the mother, whereas the vaginal birth babies are what we consider the gut microbiota. It does normalize after a while for a lot of people, but that's within the 150 years. Breast milk versus formula fed. These are great inventions, by the way. C-section is an amazing invention. Formula is an amazing thing for a mom who can't feed her baby. But it has unintended consequences of totally changing the microbiota. Diet in general is changing the microbiota, and in 150 years we've done a lot of interesting things that you guys are probably more exposed than I am about that. And then there are the drugs, and as Colleen talked about, antibiotics being an important one. But one of the things that we've done at Second Genome is also analyzed the 20 most used pharmaceuticals. These are largely for chronic diseases. Many of them are used for diabetes and cardiovascular disease, depression, things that you take, basically a pill you take every day. And what we found is that it's not just antibiotics that have an microbiome disruption. Many do. And so we can discuss that in the Q&A. So how do we take all this information, this interesting observations, and how do we translate this into drugs? This is not a complete chart by any means, and Nick will supplement this chart with something that's kind of importantly missing here. But this is just a framework to share what people are thinking about when they think about translating the microbiome into therapies. So on the far left is fecal transplants. You've heard from any talk about the microbiome, you can't avoid this. But
we've spared you.
Yes, but basically
we have great pictures.
Yeah. But it's basically a full complement of sort of normal microbiota, as you can imagine. The next type of thing, and that obviously has efficacy in C. diff, and we'll see how that progresses in terms of approvals as an actual approved therapeutic by the FDA shortly. There are companies working on what we call a consortia, defined consortia of strains that they believe have a specific action, like anti-inflammation. And so this would be the second type of interesting new drugs that you're going to, or therapies that you're going to see coming from the microbiome. You can then think about a specific strain, and actually this history is there in the probiotics industry. Both these consortia strains have been tried for a very, very thin slice of the entire diversity of the bacteria. Those bacteria that have a long history of being safe in cheese, dairy cultures, beer, those are the things that really are, that's the only one, that's like less than 1%, are the only things that have been tried in what is called probiotics or supplements. But that is an example of a strain-based therapeutic. Then you can think about something that a bacteria or a microbiome as a whole sort of secretes or sheds. So we're calling these bioactives. These could be small molecules, this could be peptides, proteins, etc. So those, if they are detrimental, then you got to think about some other way to approach it. But if they're beneficial, then you can think of them as the therapeutic itself. In the case of a detrimental bioactive, then you have to think about how does it actually affect its detrimental effect on the host. And here you can start to get into things like receptors on the gut that it interfaces with in order to do its detrimental action. And if you're able to decipher that target in the gut, then you can go after that with more conventional approaches. So at Second Genome, which is where I work, we are further to the right of the spectrum, and there are other companies that are towards the left of the spectrum, but we want to illustrate sort of the panoramic view. And Nick will talk to you about the thing that's missing, which is phage, but we'll get to that in a second. Here's a illustration, but it's actually the Second Genome sort of platform of how we go from, how we discover new drugs in the microbiome, given our philosophy to go to the right of the previous picture. So we start with a representation of human clinical disease that is as curated as possible to separate out a lot of the issues that we talked about. The noise in this signal-noise ratio here is very important. And so we start with something that represents that important unmet need in curated samples. Ulcerative colitis patients with a flare versus those that do not have ulcerative colitis, versus those that are normal colonoscopy. Where possible, we go try to get the biopsies rather than stool, but stool is obviously important in supplementary information. But you can think in certain diseases where the biopsy is the important way to diagnose the disease, like in IBD, you could start there. Then for us, it's about the challenging but important work of integrating both host biology, in this case human biology, and the microbiome side. So in the first circle, it's about understanding the microbiome. What is the community that is different in these groups? What are the bioactives that are different in these groups? So forth, to sort of ascertain both at the functional level as well as at a community level what's different. And at the bottom half of the first circle, it's about understanding how is the human side responding? What is human gene expression and how is it changing in these diseases? And integrating that information into novel disease insights that basically puts a new lens on disease. We've been really thinking about it in a way only from the human side for pharmaceutical discovery all these years. And by adding the top half of this graph, various things come alive to us that were hidden before. And from there, it becomes a job of validating, screening through the candidates that you have. And this can be done through in vitro assays, in vivo models, the fecal transplants through mice that Colleen illustrated. So if you have certain consortia of strains or bioactive, you can try to use that in the right animal models or in vitro models to try to get to functionality. So at the end, we at Second Genome really want to get to an actionable mechanism of action, preferably something that can look like a conventional therapeutic. But that's more of a business model philosophy to try to reduce regulatory and other risks, but IP risk included, which is an important issue in the field. But there is a very big world of discovery of drugs that can happen. Drugs against host targets, drugs that are based on beneficial bioactives against the microbial targets, sort of making them not shed an enzyme or increase an enzyme. And then we'll go to Nick, who'll tell you about his way of thinking through.
Sure. My name is Nick Conley. I'm CEO and co-founder of Epibiome. We're a precision microbiome engineering company. So as humans, we've become pretty adept at adding things to the microbiome. So we can culture bacteria, and then we can genetically engineer them if we want. We can add them to fermentation reactions. We can add them to foods like Greek yogurt. Now that's not to say we always understand what happens after we add it, but we can, in general, add bacteria to things. What we're not very good at, and where we really lag behind, is in the specific deletion of bacteria from complex microbial communities while leaving the rest of the population intact. So you can see in the top right, these are the tools that we have available to us. You'll note that they're all very crude and broad spectrum. They kill everything. At Epibiome, we're working to change that. So the technology that we employ are these bacterial viruses known as phages. Phages work by binding to specific receptors on the surface of bacteria. Once bound, they inject their DNA, which hijacks the host's cell machinery and creates more copies of itself. These copies can go on to infect new bacteria and kill them. So it's really like it's a form of natural antibiotics with amplification built in. And one of the nice things is these phages already have generally recognized the safe status from the FDA for inclusion on food intended for human consumption. So there's not a big safety issue here. The other nice thing about phages is that if you want to kill a particular strain of bacteria, you can actually target it with multiple phages, where each phage binds to something different on the surface and kills the bacteria before it has a chance to develop resistance. So there are some strategies here to overcome resistance, which is always a problem when you're trying to kill bacteria. So with that technology in hand, I want to discuss a really exciting opportunity that we've identified. So these are dairy cows. There are 270 million of them on the planet. These look pretty happy and healthy. There's actually a dirty little secret. There's about half of them are afflicted with a disease called mastitis in at least one of the 4 quarters of their udder. So mastitis is an infection of the udder tissue in dairy cattle. In its clinical presentation, it appears as redness, inflammation. It's painful to the touch for the cow. But for every 1 clinical case, there are actually 15 to 45 subclinical cases. So these are the cases where the cow is still infected, but it shows no symptoms. The problem with mastitis is that the bacteria infect the milk-producing tissue of the udder. So the cows make a lot less milk, and the milk is lower in quality. You can see in that cup on the left, that's what comes out of a cow with clinical mastitis. It's yellow, it's basically pus. It's got a lot of the cow's own white blood cells in it. And there's actually about a tablespoon of that in every gallon of milk that you buy at the grocery store. So that's why I drink my coffee black. So we can look at the economic impact of this disease, and it's considerable. It's about $2 billion in losses every year. In the US alone, $35 billion in losses globally each year. Most of the losses come from the fact that the cows make less milk, often over the rest of their productive lives. So that segment of the losses is shown in blue. Sometimes the milk has to be thrown away. So it's all pooled together in the milking process. And if it has too much of that pus inside of the milk, they just have to discard it. There are actually regulations that define how many of the cow's own white blood cells can be present for the milk to be sold on the market. In the green sliver, those are losses due to the fact that chronically infected animals are often just culled. They're sent to slaughter so they don't infect the rest of the herd. And then finally, that yellow slice is due to treatment and prevention of those costs. So the reason that cows get mastitis, it's the #1 reason to give them antibiotics, is they're milked 3 times a day using this really powerful vacuum milking equipment. So the milking equipment very quickly gets the milk out of the udder, but as a result, it leaves the teat canal dilated for about 2 hours. So the cows are milked 3 times a day. After each time the teat canal is dilated for 2 hours, this is 6 hours where the cows are especially susceptible to getting mastitis. When that happens, they're treated with small molecule antibiotics. You can see an intramammary infusion in the lower left, a veterinarian shooting some antibiotics into the udder through the teat canal. And in Epibiome, we want to replace these small molecule antibiotics with our phage technology. So we really think now is the right time for this. The technology is here. We've got next generation sequencing that really allows us to enable accurate identification of exactly what bacteria we're working with, what viruses we're working with. We can use this technology to understand where the viruses are binding to the bacteria. And even as few as maybe 5-10 years ago, what we're trying to do would have been cost prohibitive. In addition to the technology, we also have a lot of consumer awareness. So everyone knows about the Human Genome Project. People are starting to learn more about the Human Microbiome Project. And a lot of people are willing to accept that there are some good bacteria out there. It's not really a far leap to imagine that there might be good viruses as well. There's a huge push to stop using antibiotics unnecessarily. You saw Tyson Farms just come out, it was earlier this week, announcing that they were going to stop using antibiotics in their chicken broiler production. Sanderson Farms came out and said, we are not going to stop using antibiotics. We don't think the technology is ready yet. But it shows that it's still very early days. And it's the time where these technologies need to come into development. By 2050, we anticipate that the number of deaths from antimicrobial resistance will exceed those from cancer. And that's astounding. Today, maybe some of us, probably not many of us, know someone who's died of an antimicrobial resistant infection. We almost lost a senior scientist that we were working at Illumina just a month ago from a terrible staph infection. He survived. He was able to keep his leg. But it's this sort of thing that we're going to hear more and more about in the future. And that's why you're starting to see a lot of regulatory momentum to control antibiotic use and to come up with strategies. So the White House just released this 5-year plan on combating antibiotic resistance. The FDA announced an initiative to get antibiotics of human importance out of agriculture, especially the prophylactic, the preventive use of those antibiotics, to save them for human medicine. And the EU announced a very similar initiative, and phages were actually part of their strategy. So, turn it back over to Steve. We can start the Q&A. Looks like it's already.
Yeah, can you clarify something? With your phage therapy, you're still killing the bacteria, so you're still disrupting the microbiome. So if it was the antibiotics that was killing them, now you're killing them with the phages. So aren't you just changing one thing for another?
Yeah, so the mechanism by which phages work actually give them a considerable amount of selectivity. So they bind to very specific receptors on the surface of bacteria. What we often see are that with a particular phage that we discover in nature, it can detect, it can infect and kill maybe a few strains of a particular species. If we know that that species doesn't belong, for example, we have no reason to believe that Staph aureus actually belongs in the teat canal of the udder, then we can try to combine phages and expand our host range so that we can infect all Staph aureus, or 90%. So it really is an exquisite level of selectivity. Our Staph phages don't kill E. coli, they don't kill salmonella, they don't kill pollinizers or commensal bacteria, and we can actually do screens to demonstrate that. So if we have a community, we often use mock communities. We can use a phage, take out that bacteria that we wanted to kill, and then sequence. Just do 16S RNA bacterial profiling to show that the rest of the community is still intact. And that's there's some other experimental steps there to make sure that we're only looking at viable bacteria. But it does have that ability to make it much more of a smart bomb than a nuclear bomb.
From a publication perspective, I know researchers from companies like Bayer and Amgen have come out and said that more than half of publications aren't reproducible. Is that something that I guess that microbiome research does a better job of, making sure that that research is more reproducible? And I guess accordingly, do you see more funding going that direction? If so,
I think one of the things that the Human Microbiome Project tried to address is exactly that. So the scientists from different academic institutions, when they came together to do that project, first put together some very strict protocols around how samples are collected, how they're processed, even which sequencing technology was used and how they were analyzed, and then have created these open source areas where everyone can dump their data into alongside what protocols they used. And a lot of people have sort of stuck with those protocols just because it allows you to actually combine data and compare apples to apples. So in that sense, there is a real effort driving towards making sure that data is reproducible just because the way that it was created is being reproduced, which is a big problem in trying to reproduce data in general in science. So I think the funding question is a maybe slightly different one. I think that the government has funded now a second Human Microbiome Project, and I think that funding for the microbiome is coming because the kind of data that's been collected around the influence that the microbiome can have on a host disease state is starting to become more and more compelling across a wider variety of diseases, and so I think the funding is coming from that promise.
Question for Nick and Colleen. Is the macrophage approach a single bomb that's good forever, or is it, as with an antibiotic, basically has to be redone if the infection reoccurs?
So there's a few ways to address that. So bacteria can gain resistance, and that's something that we can combat with the cocktails, as I mentioned before. And we can also design our cocktails so that we can make resistant bacteria, but that they're no longer virulent. Once the host is eradicated, there's really no reason that the phage will be propagating anymore. It appears that in raw milk, the phage according to literature, is no longer detectable after about 3 days. That said, there are entire herds of animals that appear to be resistant to certain bacteria. And then when they go and look at the soil or sample the animals, they find that they carry a phage that appears to confer that resistance. So it's some mix. There might be some latent benefit to having that phage, in the soil, just having it around. But we don't know. I think that's the answer.
The other question for Colleen was, obviously they have Activa and probiotics and things like that. Does that only perturb the system for a short time, or can you actually change the microbiome and the whole system by taking that probiotic?
Well, I think a lot of the probiotics that are on the market today, there's a couple of questions. The first is, do they even get to your gut? So that has actually yet to be answered. And when they get to the gut, are they active? Are they performing any activity? And so, and those have even been, there was a study that came out recently out of New York in which they showed that the bottle didn't even have what they said they had in it. So there's a lot of, I think, issues with the probiotics and the yogurts that are in the space right now. But I think in an ideal scenario, being able to take either a single strain or a consortium of strains and create a change in the ecosystem of the microbiome is going to require that you can get it to the target place, the gut, and that it can be active. And there is some data coming out from some other companies in the space that this can be done. So I think Series Health and Vedanta are both using the concept of a consortia of microbiota in order to change the gut microbiome. And at least in the clinical, and Open Biome is another group that's been doing that through the so-called poop pill. And so what you can see is that these guys have been able to affect a change in the microbiome. How sustainable that change is unclear. So in the case of C. diff, you can get a microbiome change that lasts for quite a while because you're going into a system that has very few microbes in it. In a microbiome that's got a healthy ecosystem or has an ecosystem, I think that's a big challenge. I mean, that is the question.
And just to add on to that, a lot of this species, Acidophilus, so on, like to grow below pH 5, but not necessarily a lot lower than pH 5. Whereas stomach pH is somewhere between 1.5 and 3.5. It's about 0.5% hydrochloric acid, about 5,000 parts per million. So the pH scale isn't linear; it's a log scale. So we're, the reality is that a lot of these organisms actually aren't surviving well into the stomach and going further into the gut. And I'm a chemist by training, actually at a chemical consulting company. And one of the projects I worked on was understanding whether or not some commercial products, I can't get into details, were likely to remain viable into the gut. And the data were pretty common, yeah. So a lot of there was almost nothing viable into the gut. Now there are ways around this, tricks around this, and some companies are making progress there. But that is one of the things to think about.
I do find Jamie Lee Curtis very convincing in the activity shelf.
Go.
Can you come across proton pump inhibitors and their ability to affect the flora?
Yes, so one of the chronic drugs that we tested was a PPI, and it does affect it. So if you will...
So, PPI...
proton pump inhibitor. Yeah. It's like a
Prilosec. Yeah.
So if you put it on a scale where 1 is like water and 10 is a powerful antibiotic, PPIs are around 3.5 in our test.
Scott? Oh, good.
Any comments about chronic aspirin, non-steroidal use, or Tylenol use in its effect in the gut microbiome?
We actually didn't find much, so that was in the 1.5 area.
Just what the connection between the microbiome and epigenetics?
Can you repeat the
This is
question?
a company called with the word Epibiome.
Yeah.
That's what we do.
Yeah, I mean, I think that we're just starting to learn in that space, and there's going to be a lot more stuff that we learn about. So, bacteria methylation is something that exists, and mostly we believed it to exist for the purposes of replication. But I think that, you know, much like in the human genome, we'll find that there are probably some changes in expression that are linked to that. So, that's a big unknown. I mean, I don't.
Yeah. So I know Shankar Balasubramanian, the guy who invented the Selecta technology that was acquired, became Illumina's technology. I saw him speak at an ACS meeting about 6 months ago. He was winning the Tetrahedron Prize for creativity in organic chemistry. And he says that he now has reagent sets for epigenetic sequencing. So instead of your ATCG, you'll actually get the epigenetics. It says it's compatible with the Illumina's technology. And I think once that comes to market and is readily available, people will start to understand those connections. Because they will be, I think that they will be very interesting.
What was the name of that doctor?
Shankar Balasubramanian.
And you're
Well, right,
just
right
come
down
up afterwards.
Yeah.
Thanks.
Over there, please.
Can you say a little bit about how you interact with you know, foundations or the academic sector to further your companies? You know, how do you, what are effective mechanisms to interface on those fronts?
I think in this space that's really important. Most of the work has been done in academia, and we are now at the very beginning of being able to try to translate some of that into these companies. And I mean, we just recently had a translational meeting in Boston, and you could fit everybody into a room who's doing this right now. So definitely that connection to not only academics, but also clinicians is really important. So, I mean, there's a few different ways. I think everybody here has on their advisory board, certainly people from academia, who are playing significant roles in driving how we think about the microbiome and how we think about interfering with the microbiome. And I think certainly the concept of industry funding academic research in this space, if everybody's been doing it, certainly I think most people are involved in that. So, I mean, the other important part in dealing with foundations is that because the microbiome is believed to impact so many different diseases, I think there's a lot of foundations that are now becoming interested in the microbiome and interacting with both academics as well as industry on that front. There's a lot of room for collaboration.
Yeah, in particular, I mean, I used to be the director of research for the Canadian Colitis Foundation.
Oh yeah.
So I'm just curious whether you know that would be an obvious focus. You mentioned colitis.
Right. So we have actually one of our key discovery samples is from Canada. We haven't talked about that before, but I'm glad you brought it up. So yeah, those foundations, and especially in other countries like Europe and Canada, where they've really had a lot of data around these patients because of a different type of healthcare system, we really appreciated the curation of those sample sets for discovery purposes. The one other point I would add is that at Second Genome, perhaps sometimes to the chagrin of our venture capital investors, we have offered our technology to a certain type of academic or foundational or non-profit type of scientists. There are two types really, broadly speaking. For us, the cognoscenti and the microbiome that are in a core lab that do microbiome studies and involved in the microbiome project, we probably don't have much to teach them. But they're pretty much a lot of the world who are great in, say, immunology, great in rheumatology, great in whatever disease that they're thinking about, even autism, CNS areas, where they're exploring the microbiome and they really need very competent, cutting-edge ways of just getting the work done. And so at Second Genome, we've offered that to them, and that's a very important growing area for us in terms of academic collaborations.
Your mouse experiment, did they find a specific strain that resulted in the weight loss or thinning, or was it not investigated?
Definitely investigated.
Yeah.
So, and investigated across multiple groups looking at the same thing. And investigated by multiple companies, I think, at this point, too. I think to kind of speak to Mohan's slide, some of the things that people are starting to realize is that the functionality of strains needs to be understood and tied to the strain itself. And in different ecosystems, these different bacteria do different things. And so I think that's still being worked out, but there's been a hefty amount of literature trying to get at that exact question.
Okay, so I had a question.
which is, what are you doing today to improve your microbiome?
Not taking antibiotics unless I have to. We actually don't.
And your family.
And my family, yeah. We actually don't have any antibacterial soaps in our house or antiseptic wipes. I don't think it's necessary to be so crazy about everything. And I think that the other thing is I let my kids not wash their hands I'm not a big fan of Purell. We don't do Purell in our house. But I think the idea here is striking the balance between, not letting harmful microbes, because there's definitely harmful bacteria and viruses that we know about, and trying to maintain an ecosystem that is normal. So particularly for children, I have 2 kids, letting them play outside, letting them hug the dog, letting them be part of their environment. This is all helping them, exposing them to things that later they could develop allergies to is important early in life, rather than trying to keep them in a septic, sterile environment.
Is there much research among the spike about the changes in the microbiome with aging, irrespective of nuclear blast from antibiotics? I mean, just the natural aging process.
Yeah, as you age, the diversity is less. So that's the major finding. I think there are other subtle findings. There's definitely older people have less diverse microbiome, and less diverse is generally a bad thing.
And you were going to...
No, I was going to answer your question about personally.
Yes.
I think
Yes.
we were going down the line.
Yeah.
But anyway, we definitely have a dog. That's, I think, an important thing for us to learn. I think that while it's hard to avoid these antibiotics, particularly when you need it, I really take judicious care about whether we should administer it in our family. But then I follow it up almost to like 3 months time frame with whatever I can find. Even though the probiotics, I know I've even done a test ourselves in our own company, they don't quite work, but that's the best thing we have. So I do eat a yogurt, I do eat cheese, I drink a lot of beer, whatever I need to do to get some microbes in me after a post-antibiotic, or the kids is a harder thing with beer. But anyway, but you get the idea. It's an important thing to do. And then last, with the advent of new technologies, there are companies now that are offering consumer microbiomics. I have my own personal opinions about the veracity of all the data and so forth. But where I do use them is when I go through a episode. So for example, if I'm traveling, say to India, or I take an antibiotic or do something, I do a pre and post. It's a little bit of money, but for me it's being open, it's really changed. I wouldn't have thought about that was I not working at a microbiome.
Yeah, so speaking of being born cesarean section, I was, and I've actually had a fair amount of atopic disease, so allergies, rhinitis, eczema. I hold a clinic record for the maximum, they do the allergy skin tests. I think I test positive for everything. Environmental, not foods, fortunately. So, had I been able to give my parents instructions, I might have suggested inoculating me with my mom's vaginal secretions shortly after cesarean section. That's apparently a fairly common thing that some OB and GYNs are doing now. Might be something to think about. I don't know if it will work or not. Probably can't be too terrible.
It's better than the hospital wall, which is what the publication shows. The hospital wall bacteria are what's shown in the publication.
Yeah. Other things, I drink kefir. I like it. I think it tastes good. I'm lactose intolerant, and I have a lot of success with Greek yogurts, kefirs. These lactobacillus love to eat the lactose. And what else? Yeah, I don't try to stay super clean. If you're going to use soap, wash the parts where you have sweat glands, and maybe not pay as much attention to those that don't. So, guys, if you've got a beard, you actually do have to wash this. Armpits, groin area. But do you really need to wash everywhere? And then pay attention to your detergents. As a formulation chemist, some of them are really, really harsh. You probably shouldn't even be using them on your skin. So those are some things to think about. Probably won't change your life, but I think we will learn more with time. You know, for those of you who are curious, Biome, another group with which we're all familiar, they made the New York Times for that. It's an ammonia-degrading bacteria that you can spray on and use it in lieu of soap. I think they're actually working on a soap now that's compatible with it, so that you can still practice some of your normal hygiene protocols. But you know, I think you're going to start seeing more stuff like that. It's really neat, and it really invites a lot of citizen science. So, you know, what works for you? Do an experiment. You know, don't do anything that's potentially harmful. But you know, with Biome, another company, they'll sell you a kit where you can sample from pretty much anywhere you want on your body. They'll send it back to you with your microbiome in that area. So that's a good chance to play around, see what a new diet does, see what a new routine does, and you know, don't take any of it too seriously. But it's fun. It's fun to learn. It's fun to experiment.
A question.
A follow-up question. There seems to be this proliferation of probiotics on the marketplace in the nutraceutical space that you can just go buy, whether it's a drink or otherwise. And I'd like the panels, and they seem to be measured by, you know, how many colony-forming units they have within them rather than what they'll actually do to you. I'd love to have the experts' opinions on, you know, what is that all about, and should we be drinking that stuff?
And can I add, and where is it going? You know, like 3 years from now, where do we think, or 5, some reasonable time frame?
Yeah. I actually wanted to hear my fellow panelists' answers on this, but what we did do an experiment in-house where we took the common probiotics, and mostly we found that the dosage forms are not really there, or they're not there at all. So that you can take that for what it will. But the answer, for me, a piece of the answer to your question is that this thing, with the microbiome, has gone from, if you will, the lab bench to the kitchen table in record time. If you think about just genomics, if you think about proteomics, even I mean, I've never talked about proteomics at my dinner table. But in any case, but something about this captures the fascination of people. It's on the front page of the New York Times Magazine. There's something about this that grabs people. And I think that is the trend that probiotics is riding on, even though it's clearly spotty efficacy or whatever. The other point I would make, and I'd like to hear other people's opinions on it, is that this speaks to me of a personalized medicine response. In other words, there's some people who probably do respond to the lactobacillus and acidophilus, and maybe they respond at different times in their life, perhaps. And so but we're trying to get this universal big response. We're not getting it. And plus, by the way, I do think there's a placebo effect, and I'm okay with that for myself.
Yeah, I think I will say that I don't think that the science has driven the probiotic industry, but maybe almost a little bit the other way around. So probiotics have been around for a lot longer than we've been able to do science behind the microbiome. And here in the US, we think of this as being this totally novel thing, this concept that there are good bacteria. But around the world, that's not true. A lot of people have very different hygienic practices than what we have here, and aren't so, don't, like, as Steve was saying early on, don't look at the microbes around them as terrorists that they have to live in fear of. So I think that the probiotic industry is big because it's really big in Europe, and it's big here in the US. It's big because, as Mohan said, there are believers. It does work for some people, and I think right now what we're all trying to do is to make it a more refined science. So, an individual's gut microbiome is not going to respond the same as the next person's, and so I think what we're going to find as we move forward, to answer your question, is there going to be more refined methodologies in which we can change the microbiome for specific groups of people, and that's really at the heart of what a lot of these microbiome companies are trying to do, is to just get a little bit more science behind it and a little bit more specificity.
Yeah, and then let's say take a look at the Mayo Clinic's webpage. They actually assess different strains of probiotics and show, for what claims are there evidence, for what diseases, indications is there evidence of a potential improvement, and which are counterindicated. That's one thing. Also, look at the other stuff, right? Because it's not just the bacteria. Look at something like Yakult. It's got an enormous amount of sugar in it. So do you really want that just to get your whatever billion dose of bugs. And yeah, those are the 2.7 ones me.
Any other questions?
Well, thank you very much, Steve and all the panelists for being here.